Hey, this is Dr. Fry, and this is the second lecture on Chapter 6, which is reproduction at the cell level. So the last one, we really went over the cell cycle and the stages of mitosis. And this presentation is really when things go awry in that cycle, it's actually one of the main causes of cancer. Okay, so at the end of this, you should be able to outline the three main checkpoints, which regulate cell division, identify internal and external factors which control the cell cycle, describe apoptosis, relate uncontrolled cell division to the formation of tumors, is cancer, identify the two types of tumors, define and give examples of carcinogens, and describe how cancer is treated. That's the vocabulary. All right, so we're going to first start out with controlling the cell cycle. So the cell cycle is controlled by using information from cells' internal and external environments. This allows the cell to determine if and when cell division is appropriate so that it does not waste resources and energy. And it doesn't begin cell division when conditions are not good. Okay, for instance, cell division would be inappropriate if the cell's DNA was damaged or there was no room for more cells in a particular tissue or organ. So all the right things have to be turned on at the right time, and things have to be turned off at the right time. That's the idea. The cell cycle has checkpoints which determine whether the cell cycle should proceed. There are three main checkpoints which occur at the transition points between phases. And here is the three checkpoints. So here is checkpoint 1. So this is at the transition between G1 and S phase. So this is going to make sure that you have enough of nucleotides and everything else to actually, this is where the DNA gets copied, right? That's what we did last time. Checkpoint 2 is a transition between G and mitosis. And this is really going to make sure that you have everything set up to actually begin mitosis. And then you have a checkpoint between metaphase and anaphase. And this is sort of to make sure all the, that's right here. So metaphase is when they line up in that middle plate. And so they have to make sure all those chromosomes are actually lined up. All right. So the four criteria that most cells must meet by the end of G1 phase. So G1 is down here just before you start DNA synthesis. Is the cell large enough to undergo cell division? Does the cell have adequate levels of nutrients and energy reserves to complete cell division? Is the cell receiving molecular signals, which could be, ligands or, outside cues from other cells in the tissue that cell division is appropriate? And DNA, is the DNA intact with no damage? So you don't want any mutations in your DNA. So these criteria may change depending on the type of cell. Once the G1 checkpoint criteria has been met, the cell can enter the S phase, and that's down here. So the S phase is when you replicate your DNA. That's when you have that leading and lagging strand and the Okazaki fragments and all that stuff. So cells which do not meet these criteria may exit the cell cycle and enter a rest state called G0. That's this right here. And they will not go through this cell cycle. So they'll just be a regular cell. They're not dividing. So these cells may return to the cell cycle once all criteria have been met or stay in the G0 phase permanently. So I think I told you before, some cells divide all the time, like your hair cells or your fingernail cells, or your skin cells, but something like your bone cells or things like that, they don't divide as often. So they spend a lot more time here in this G0 phase. Okay, so now we're going to the G2 checkpoint. The G2 checkpoint, the cell has already gone through DNA replication. And so now there are two criteria for this checkpoint are, is any part of the DNA molecule damaged? And was the DNA, molecule completely copied during the S phase? So they're really checking whether the DNA got replicated correctly. Okay, so this is the G2 right up here, right? So here's G2. So once both of those criteria are met and the cell will proceed into mitosis, if the damage to the DNA molecule is detected, then the cell will pause until the cell's repair mechanisms can fix the damaged DNA. So it is checking for mutations and damage. Okay, so sometimes the DNA is too damaged to be repaired, and this causes the cell to undergo programmed cell death, which is called apoptosis. So apoptosis prevents any further daughter cells from containing the same damaged DNA, which can impair the functioning of the cell and cause diseases like cancer. So you really, if it's something so damaged, you need to get rid of it so it doesn't keep copying itself, which is what happens with cancer. Okay, so now the final cycle checkpoint is this occurs partway through mitosis. Here the examines whether the sister chromatids are correctly attached to the spindle fibers, right? So this is metaphase right here. So this is where they're lined up. And so sometimes you can have like both sister chromatids come over here and none will come over here, right? So this is really making that they line up correctly and actually that one sister chromatid goes here and one sister chromatid goes here. That's what this is checking for. So the check occurs before the spindle fibers pull the chromatids into opposite poles of the cell. This checkpoint is sometimes called the spindle checkpoint. Okay, so I'm going to explain why the cell needs to regulate the cell cycle. The cell cycle must be controlled to ensure that cells are only undergoing cell division when it's appropriate to do so. So you want to get rid of any mistakes. Name two conditions that must be met before the cell can move through all the cell cycles. So they have to have adequate nutrients, energy reserves, sufficient space in the organ or tissue. Here are the three checkpoints. Name the three main checkpoints in the cell cycle and give one criteria that a cell must meet for each. So there they are. Describe what the G0 phase is and when a cell enters it. So a cell may enter the G0 phase if it fails to meet any of the criteria from any of the checkpoints. This is a state of rest which allows the cell to pause until the criteria for the relevant phase has been met. Okay, now we're going to talk about factors that control the cell cycle. So you can have external factors and you can have internal factors. So external factors, things that come from the outside, so often involve the cell communicating with both neighboring cells as well as those in distant parts of the organism's body. So this is sort of the different organs talking to each other. External factors can be further classified into two types: physical signals such as cell-to-cell contact, and chemical signals such as growth factors. So here you have, so if you have a positive growth factor, the cell will divide. If you have a negative growth, if you don't have the growth factor, the cell will not divide, right? So here is a cancer cell. This growth factor really doesn't have any effect, and the cells are just going to divide anyways. Okay, so so these are normal cells, right? So the normal cells are listening to the growth factors. The cancer cells do not listen to the growth factors. They do what they want to do. Okay, once the external factors have made links with the receptors on the cell surface, Internal factors are then stimulated. Two examples of factors which are found in eukaryotic cells are kinases. So these are the things that get phosphorylated and turn things on, right? So it's going to receive a phosphate group from ATP, and I think I showed you this with that kinase phosphorylation cascade. I showed you that before. But basically, this is a protein, and then when it gets this phosphate from this ATP, it actually turns it on. And then this was actually going to meet up with this cyclin. This is a different protein. And these are molecular signals which can tell the cell to move through the phases of the cell cycle. So these kinases and cyclins actually work together. And I will show you that right here. So here is a kinase, right? And here that kinase got phosphorylated from ATP. And so when it gets phosphorylated, it's going to meet up with that cyclin. And that cyclin is going to have a target protein, and that phosphate is actually going to activate this protein here. And then when it activates that protein, this cyclin is going to disintegrate and go back to its parts, and then can be recycled back again. But here is the cyclin bonds and activates. This is the kinase right here, the kinase, and then the kinase activates target protein to regulate the cell cycle, and then the cyclin is destroyed down here. And there are different types of cyclin. So here's the cyclin for G1 that actually is active during the whole cell cycle. Here's G1S cyclin, here's S cyclin, here's M cyclin. So these are all different versions of cyclin here, and they're specific to, this one is just specific to the G1S phase, right? This is the gaps, right? Here's the gap, and here's a gap, and here's a gap. And so they're active during certain parts of the cell cycle. Okay, so so it helps the cell to identify which cells need to grow and divide and which cells need to die through apoptosis. So apoptosis is programmed cell death. So it's triggered when the cell's internal or external signals stimulate genes that can help the cells to produce self-destructive enzymes, which would be like lysosomes. And here is the cell is beginning apoptosis, the formation of bleebing, right? And here's bleebs here. These, you could actually call these kind of lysosomes. And then there are partition of cytoplasm nucleus in apoptotic bodies. And then here is, they're just phagocytosis. They're just sort of bleeding this out, right? So the whole cell is just sort of disintegrating. Okay. So apoptosis allows the body to remove non-essential tissues, such as the webbing between fingers and toes. Also occurs during the loss of a tadpole's tail as it becomes a frog. So that's this right here, right? And here's why we don't have webbed fingers or feet or toes. And it is responsible for the loss of brain cells called neurons as connections within the brain are formed. So apoptosis is actually important. Okay, so we'll do this comprehension text. So describe and give an example of the two types of factors which control the cell cycle. So there's external factors such as signals from other cells and internal factors from the growth factors within the cell. Describe kinases. They are chemical signals which assist with the utilization of ATP energy in the cell cycle. So basically, they get phosphorylated and then they turn things on, is what they do. And then when the phosphate comes off of that kinase, the protein gets turned off. So what describes cyclin? A cyclin is a chemical signal which tells the cell to process through the phases of the cell cycle, or it starts it or stops it. And apoptosis is programmed cell death. Okay, uncontrolled cell division. There's where we're going to talk about cancer. If the cell division isn't controlled, what happens to your body? Uncontrolled cell division can lead to cancer. Cancer is the most common name for a class of diseases characterized by uncontrolled cell division and happens when the regulation of the cell cycle breaks down. So again, this is the one that showed you before. So this is normal. So normal cells listen to the factors. Cancer cells do not listen to the growth factors. So unlike normal cells, cancer cells grown in a culture dish continue to divide even when surrounded by neighboring cells. They continue to divide in the absence of many of the growth factors required for division in healthy cells and are able to ignore many of the checks and balances that the body has in place. So it doesn't listen to those checkpoints. As a result, these cells divide much more frequently than healthy cells, forming disorganized clumps called tumors, right? So this is a normal cell. So if you have cell damage, basically it's going to kill itself, right? So this is apoptosis, it's going to die. But here, these cells, so here's the first mutation, right? And now this cell is going to divide. This is the second mutation, and it's still going to divide though. And now here's a third mutation, but it's still going to divide. And now here's the fourth mutation. And this fourth mutation is where you get this uncontrolled growth. So it can be from one mutation, it can be from two mutations. It doesn't have to be from four mutations. This is just an example. But then you have this uncontrolled cell growth. Now it's not going to listen to any of the checkpoints or cues it's going to get from the cell cycle. Okay, so the table below shows differences between healthy cells and cancer cells. So normal cells are usually small, uniformly shaped nuclei, relatively large cytoplasmic volume, right? So this is the nucleus. Cancer cells are large, variable shaped nuclei, so the nuclei aren't round, and relatively small cytoplasmic volume, right? So this has a bigger nucleus, and it's oddly shaped, and the cell's oddly shaped. Many possess differential cell structures, normal presentation of surface markers. Those would be proteins, like receptor proteins. And this has loss of normal specialized features, elevated expression of certain cell markers. And that's how they sort of tell if you have cancer, by those markers. Conformity in cell size and shape, shells arranged in discrete tissue, so that's here. Variation in size and shape, disorganized, arranged in cells. So these are much more organized, these are much more disorganized. And this says lower levels of dividing cells, cell tissues clearly demarcated, and large number of divided cells, poorly defined tumor boundaries. Right, so that's the idea there. Okay, so comprehension check. So the cells in the image below show two different groups of the same type cell labeled cell group A and cell group B. Decide which of these two cell groups show healthy cells and which show cancerous cells, right? So these are healthy. These have more cytoplasm, a smaller nucleus, they're regularly shaped. This one is irregularly shaped, it has a long nuclei, they're clumped, they're not organized, things like that. Okay, so I'm going to skip over that. Okay, so categorizing tumors. So tumors can be categorized into two main groups, benign and malignant. In benign tumors, the cancer cells usually remain collected together as a mass. The tumor may be relatively harmless and can usually be cured by removing it during surgery. A malignant tumor, some of the cancer cells break away or spread from the tumor, right? So, and they can spread to other parts of the body. These breakaway cells are carried in the bloodstream or lymphatic system to other parts of the body where they can form tumors called metastasizes. Once a tumor metastasizes, it is a lot more difficult to completely remove it from the body. So that's when you get to stage 3 and stage 4 cancer. So that's invasive, right? So this is where it's intact, and this is where it's not intact, right? So malignant cancers form a mass of cells which are contained in one area. That would be benign, so that's false. Benign tumors can often be removed and therefore cured. That is true. Metastatic cancer is harder to treat than benign cancer. That's true. And malignant tumors use the bloodstream and lymphatic systems to form metastasis. I don't know how to pronounce that. Okay, the cause and effect of cancer cells. So cancer cells are normal cells that have suffered damage to their DNA. When one of these cells incur DNA damage, the cell no longer is able to make protein, or alternatively may begin to make a different protein, which encourages the cell to multiply uncontrollably. So we've seen that before. Most cancers can carry mutations or errors in one or more of these three types of genes. So oncogenes, they speed up the cell cycle and turn healthy cells into cancerous ones. Tumor suppressor genes ensure that the cell does not divide when it is not supposed to. So damaged DNA is repaired and apoptosis is carried out. So this is actually a good thing. This is a bad thing. DNA repair genes fix mismatched DNA pairs. So this is going to fix the mutations. Okay, the cause and effect of cancers. So individuals may inherit a mutation in these genes from their parents, and these are called germline mutations. So examples are those breast cancer genes, right? And I think you can actually get tested. So if you have breast cancer in your family, you can actually be tested for that. And a lot of people have breast mastectomies before they get breast cancer. And this is an example of that. So this is normal. And then this is breast cancer. Okay. So can define the term germline mutation, a mutation which is passed on from one generation to the next because it occurs in the gametes. So you get that from your mom or your dad. And give an example of a germline mutation which causes breast cancer. And name the three types of genes that are known to play a role in cancer. And that would be those three right there. So not all cancer-causing mutations are inherited. These mutations are called acquired mutations. Acquired mutations are the result of being exposed to radiation or chemicals, which are known to cause cancer. So substances that have the potential to or are known to cause cancer are called carcinogens. Examples of carcinogens include UV radiation, so that's why you should wear sunscreen, right? So when we had the ozone layer was depleted, right? So more UV radiation got through to we received more UV radiation, and that increased skin cancer. Everybody knows that you shouldn't smoke tobacco or vape. That causes cancer. Some viruses cause cancer. So the other one I'm going to put here is this PFAS, or it's also called C8. There's actually a documentary. It's actually not on Netflix anymore. It's on YouTube. But it is called The Devil We Know. And these PFAS they cause all kinds of cancer, right? So testicular cancer, ovarian cancer, prostate cancer, kidney cancer, non-Hodgkin lymphoma cancer. I don't think I can show you this video here. The sound doesn't come through. But this is the trailer, and I think I am going to have you watch this movie. It's about DuPont. It's in West Virginia. So what they have done is they make this 3M, and they make these like Teflon is one, and like anything with Gore-Tex in it, like anything waterproof, or like Teflon pans you cook with. that all has PFAS in it. And actually, the factory is in West Virginia, and they have put all their effluent in the Ohio River. Well, we are actually downstream from that. So that water actually flows out of that plant into the Ohio River, and it actually flows through Louisville. So you probably have PFAS in your blood. So almost everybody has it in their blood. I think I'll have you watch that. I'm going to assign that movie next week. Okay, so effects of cancer. It stops the body from performing the specific functions needed for it to remain healthy. If cancer cells continue to grow unchecked, they will eventually kill the organism. So, for example, if you have lung cancer, you will no longer have the gas exchange. So you need to take in oxygen for a cellular respiration, and then you breathe out carbon dioxide. In the brain, electrical messages fail to be interpreted correctly. In the skull, restricted space, so some regions will be unable to function properly. So, treating cancer, there are two main forms of treatment for cancer. There's radiation therapy, and that damages the cancer cell's DNA using localized radiation. The problem with that is it doesn't just damage your cancer cell's DNA; it also affects your other cells as well. But so it has to be very localized, right? So it's very localized. None of this stuff is very good for you. And then chemotherapy is actually drugs which travel through the whole body, which kill actively dividing cells. And again, this can not just affect your cancer cells, but it can affect other cells as well. Like a lot of people lose their hair, right, when they do chemotherapy, because your hair cells are dividing. All right. So, medical regulation of the cell cycle in cancer. So, medical researchers use laboratory grown cancer cells in the search for cancer treatments. The most famous cancer cells used for research is called the HeLa cells, and they obtained in 1951 from a cervical tumor removed from a woman named Henrietta Lacks, right? So that's where you get the HeLa from. The cell lines continues to be grown and studied in laboratories all over the world. Okay, so an acquired mutation can be inherited from your parents, whereas a germline mutation cannot. So that's false. A germline mutation is what you get from your parents. Carcinogens have substances that are potential to cause cancer. That's true. Tobacco smoke, petrol, and alcohol. So if you're an alcoholic, a lot of times you'll get liver cancer. Are example of, I don't know what this is, of carcinogens, and this is where PFAS would also be. Radiotherapy kills rapidly dividing cells. That's false. HeLa is cells or a strain of cancer cells used to study the treatments for cancer. That is true. And that is it. So that's the end of this lecture.